The overall goal of this procedure is to probe the dynamics of protonation and conformational changes of two photosensitive membrane proteins using time-resolved step-scan FTIR spectroscopy. This is accomplished by first forming a hydrated protein film whose infrared absorption will be probed either in an attenuated total reflection configuration for bacteriorhodopsin or in a transmission configuration for channel rhodopsin 2. The second step is to excite the sample with a nanosecond laser flash of suitable wavelength to initiate a photocyclic reaction in the protein, detecting time-resolved changes in the intensity of the infrared light interacting with the sample. Next, the above process is repeated at discrete positions of the mobile mirror of an interferometer, which controls the optical path difference between two split beams, until a complete time-resolved interferogram is recorded. The final step is to transform the time-resolved interferogram to a time-resolved IR difference absorption spectra using the Fourier-transform and the Lambert-Beer law. Ultimately, the time evolution of bands, especially in the amide 1 and in the CO double bond carboxylic regions, are inspected to obtain dynamics of conformational and protonation changes in the protein.
The main advantage of the technique over most existing experimental methods is that it resolves transient protonation changes in proteins. Moreover, it does it on the micro and millisecond timescales, the most relevant time range to probe protein functionality.
This method can help to answer key questions in the molecular biophysics and protein science field, such as which residues deprotonate and when they do during the functional mechanism of a protein.